So now that we've gone over simple or Mendelian genetics, we're going to get into a variety of non-Mendelian genetic concepts. These are concepts that expand beyond the simple dominant recessive relationship that we saw with Mendel. The first of those concepts we're going to get into is incomplete dominance. Incomplete dominance is where the heterozygote has an in-between phenotype, meaning that there's a homozygous dominant individual, a homozygous recessive individual, and the heterozygote has a trait that's in between the two. And so in this case, the homozygous individuals don't really have a true dominant or recessive relationship. So it's not true dominance. So because of that, we change how we write those alleles. So if you remember when we were looking at simple dominance, we used capital letters to show dominant alleles and lowercase letters to show recessive alleles. But in this case, we're using capital letters for both alleles. And instead, in order to tell the difference, we're using a superscript to tell us that these alleles are different. So for example, if we're looking at flowers and flower color, we might use the letter F to tell us that we're looking at the flower color gene. And then to determine the two different alleles, we use a superscript R to tell us that it's red, and a superscript W to tell us that it's white. So the single letter F tells us that we're looking at the same gene, but the superscript tells us these alleles are different from each other. So let's look at a working example. In this case, we're looking at a mother and father snapdragon flower plants that have two different colors. The mother is white and the father is red. In the mother's case, because she is white, she is homozygous for the white allele. She is C superscript W, C superscript W. So we could say she's homozygous for C superscript W. The father has red flowers, and so they're homozygous for red. So they are C superscript R, C superscript R, or homozygous C superscript R. And when we cross these two together, we end up with heterozygotes for all offspring. So every offspring has one C superscript W allele and one C superscript R allele. Now, because it's incomplete dominance, not a simple dominant recessive relationship, we see that the heterozygote looks like an in-between between the two parents. They end up looking pink, right? And pink serves as an in-between between red and white. Now, if we were to cross two pink snapdragon plants, meaning both are heterozygous, meaning they're C superscript W and C superscript R, we see that we end up with a 1:2 to 1 genotypic ratio, with one individual being C superscript W, C superscript W, two individuals being C superscript W, C superscript R, and one individual being C superscript R, C superscript R. The individual homozygous for C superscript W ends up being white. The individual that is homozygous for C superscript R ends up being red, and the two heterozygotes end up being pink like the parents. So let's do our own example of this, looking at red and blue flowers instead. And we're going to cross two heterozygotes. So these individuals are going to be purple. Their potential gametes are going to be C superscript R and C superscript B. And so if we fill in our Punnett square, as we've done in the past, we find a Punnett square that looks like this, where one individual is homozygous for C superscript R, another individual is homozygous for C superscript B, and two individuals are heterozygous. This gives us a 1:2 to 1 ratio for color, with one individual being red, two being purple, and one being blue. And so even though we only have two alleles, we see three different phenotypes because the heterozygote is an intermediate between the two homozygous genotypes. Another non-Mendelian concept is that of multiple allelism. Multiple allelism means that there's more than two alleles for a gene within a population. Remember before we were talking about Mendelian genetics, we had a simple dominant relationship, and there were only 2 possible alleles in a group. But in reality, a population can have 3, 5, 10, 100 different alleles for one single gene. Do remember that an individual will only have 2 alleles because you have 2 copies of each gene, but if you look at the whole population, you can find many different versions of those alleles. The best example is the ABO blood group in humans, where the gene has 3 different alleles, IA, IB, and i, and shows us 4 different total phenotypes using different combinations of these 3 alleles, type A, type B, type AB, and type O blood. This also introduces another concept to us, in that the IA and IB alleles are co-dominant. This is somewhat similar to incomplete dominance, although there's a major difference in what the heterozygotes look like. In both incomplete and co-dominance, we see the heterozygotes have a different phenotype than the homozygotes. However, in incomplete dominance, the heterozygote is a blend of the 2. But in co-dominance, we see both of the alleles at the same time, simultaneously, equally, and discretely. We'll talk a little bit more about that, and we went into this in our blood typing lab, so I would definitely look back at your lab for that explanation on co-dominance and blood type. But let's dive in a little bit further. So like I said, there are 3 possible alleles, IA, IB, and i. We notice that the IA and IB alleles have capital I's. This means that they are both going to be dominant over our lowercase I. But what happens if you have IA, IB, where both I's are capital? In that case, you end up with type AB blood. And type AB blood is not a blend or a different blood type from A or B, it's a combination of both. This individual will express both A antigens and B antigens on their blood cells, rather than some new in-between antigen. We also see that with 3 alleles, there are 6 different possible genotypes: homozygous IA, IA little I, homozygous IB, IB little I, IA, IB, and i. This is how we get the 4 different phenotypes: type A, type B, type AB, and type O. Type A has 2 possible genotypes for it. You can be homozygous for IA, or you can be IA little I. Remember that the IA will mask the little I. We're following a simple dominant recessive relationship there. And we see the same setup for type B. You can be homozygous for IB, or you could be IB little I, where the IB masks the little I. For type AB, there's only one possibility with IAIB, and type O has only one possibility with little I. Now, when we were looking at Mendelian genetics, we were focused on one gene giving rise to one trait. However, in the human body and in most living things, most traits we see are composed or controlled by many genes working together. We call these traits polygenic traits because poly meaning many, genic meaning genes, many genes control the actual end outcome you see. So for example, if we're looking at the genes that control how tall you end up, there's more than 180 genes that influence that. It's the combination of these 180 genes that tells you how tall you'll actually be. These traits typically have additive effects too, meaning the more of a certain allele you have, the more that trait will be expressed. It's kind of like adding logs to a bonfire. The more logs you put in the fire, the bigger the fire will be. So let's say we're looking at those height genes, and having a dominant allele means you'll be taller. The more tall alleles you end up with, or the more dominant alleles you end up with, the taller you'll be. If you end up with more recessive alleles, then the shorter you'll be. We also have the opposite of that happen as well, known as pleiotropy, where one single gene can have an effect on many different traits. There are many examples of pleiotropy, and we see this in a lot of different conditions like Marfan syndrome or sickle cell disease. But here's a good example of polygenic traits in action. You have these three gentlemen standing here, and depending on how many dominant and recessive alleles they have for height, the additive effect comes into play. The more dominant alleles they have for those 180 genes, the taller they end up, and the fewer, then the shorter they are. Now let's get into pleiotropy. Pleiotropy is one single gene that has many, many different effects. And sickle cell disease is a good example of that. The sickle cell allele, and sickle cell disease in particular, can have a wide range of effects on the human body. The main gene, though, the one gene that's controlling all these changes, is a gene that focuses on shaping hemoglobin. Hemoglobin carries around oxygen on red blood cells, and if you change the hemoglobin shape, you can actually change the entire structure of the red blood cell. And that's what happens with the sickle cell allele. It causes the red blood cell to form an unusual sickle shape rather than the nice round puffed up donut-like shape that they have under normal circumstances. It also does provide resistance to a condition or a disease called malaria. Now, if you are HbA, you're a homozygote for normal red blood cells. And so none of your cells have any sort of sickling shape, and you don't have sickle cell disease. However, if you live in an area where malaria is endemic or naturally found, then you can catch malaria and get really sick. If you are HbS, or homozygote for sickle cell, you are immune to malaria. However, you have significant sickling of red blood cells, and so you have what we call sickle cell disease. And the effects of that disease are widespread, including things like fatigue, brain damage, and even organ damage. The real winner in this situation is the heterozygote. The heterozygote, or HbS-HbA, does have some sickled cells. However, there are also healthy red blood cells alongside that. So you'd have enough healthy red blood cells to prevent yourself from developing sickle cell disease and all of those problems down the line. But because you have some sickled red blood cells, you're also immune to malaria. This is also a good example of the heterozygote advantage, where the heterozygote has a good advantage over the homozygous phenotypes on both ends of the spectrum. It's really interesting to look at pleiotropy in that you have one gene that has so many wide effects, and it's something that we see in many different diseases. It can also be why it's so hard to treat many of those conditions, because while there's one single simple cause, the fix for it is so widespread and different and diverse because of how many different effects it has downstream. Definitely take some time to review these different types of non-Mendelian concepts. These will come back on that unit exam.